Thanks for reminding me. I totally would have forgotten that. So let's get started. I got a big, fancy, complicated data set. I'm gonna shrink it down dimension-wise. I got a big, fancy, complicated data set. I'm gonna shrink it down from a lot of dimensions down to just a few dimensions. It's gonna be totally awesome. StatQuest. Yeah. All right, everybody. Thank you very much for coming to class. Let me get this. I'm all tangled up on my ukulele. Let me get this thing off me. So let's get started. I'm going to share my screen. Bam. All right. So can everyone see that?
Yes.
Hooray. Okay, so today we're going to cover 3 ways to make a big, funky, complicated data set easier to make sense of. Oops. One way is called PCA, which is short for Principal Component Analysis. Another way is called MDS, which is short for multi-dimensional scaling, which, to be honest, is the coolest sounding of these 3 techniques. And the last one is called linear discriminant analysis. They all sound super fancy and complicated. They're not. So without further ado, let's start talking about PCA. And we're going to go through PCA step by step. And we're actually going to go through it step by step multiple times. So by the time we're done with PCA, you guys will have it down cold. Okay, so in this StatQuest, we're going to go through principal component analysis one step at a time using something called singular value decomposition. So there are 2 primary ways to do PCA. One is to use something called singular value decomposition, which sounds super fancy, and we'll go through that. And the other way, the older way, is to use what's called a covariance matrix. And a lot of times when people teach PCA, they teach it from the covariance matrix perspective, but nobody ever does PCA that way. Everybody does PCA with singular value decomposition. And so that's the way we're going to do it. I actually think it's more intuitive to go through the singular value decomposition way. So it not only is it the newer way that everyone actually does in practice, it's actually easier to understand. However, just so we can cover all our bases, when we get through the end of this, we're actually going to review how to think about PCA in terms of a covariance matrix. And so you'll have both perspectives of the same process. Okay, so what we're going to do is we're going to learn what PCA does, how it does it, and how to use it to get deeper insight into your data. So we're going to start with a simple data set. We've measured transcription of 2 genes, gene 1 and gene 2, in 6 different mice. Note, if you're not into mice and genes, and I know a lot of people are not, I used to work in a mouse genetics lab, and so I used to be way into them. That was my thing. But some people aren't into mice and genes. So you can think of the mice as individual samples and the genes as variables that we measure for each sample. For example, the samples could be students in high school, and the variables could be test scores in math and reading. Or the samples could be businesses, and the variables could be market capitalization and number of employees, or whatever. It's just data. Okay, so now we're back to mice and genes. If we only measure one gene, we can plot the data on a number line. Mice 1, 2, and 3 have relatively high values, and mice 4, 5, and 6 have relatively low values. In math land, a number line is called a one-dimensional graph because a line spans a single dimension. Even though it's a simple graph and it shows us that mice 1, 2, and 3 are most similar to each other than they are to mice 4, 5, and 6, if we measured 2 genes, then we can plot the data on a 2-dimensional XY graph. And in this case, gene 1 is the x-axis spans one of the two dimensions in this graph, and gene 2 is the y-axis and spans the other dimension. We can see that we can see that mice 1, 2, and 3 cluster on the right side, and mice 4, 5, and 6 cluster in the lower left side. If we measured 3 genes, we would we would add another axis to the graph to make it look 3D or three-dimensional. So in this case, we've got smaller dots that have larger values for gene 3, and they're further away. And the small dots there's a miss. Smaller dots have larger values for gene 3 and are further away. The larger dots have smaller values for gene 3 and are closer. And this is something, I've got this video online and people watch it all the time. And this is something that people are perpetually confused with. In that I'm trying to draw something that's three-dimensional on a two-dimensional surface. Your computer screen is only 2 dimensions, but you just imagine that there's depth, and imagine that gene 3 goes out the back of your screen and into the wall behind you or out the window or wherever you're sitting. It just keeps going. And so it represents some sort of depth, and that we're just sort of trying to fake it on this two-dimensional screen of yours. Okay, if we measured four genes, however, we can no longer plot the data. 4 genes require 4 dimensions. And yes, a lot of people say, hey, what if we change the shapes of things? Couldn't that be represent a dimension or something? And that's true, or we can change colors, and there's lots of little things you can do to kind of fake extra dimensions. But the reality is, if we have lots of dimensions, and oftentimes with genomics data sets, we've got 20,000 dimensions. We might have measured every single gene in the mouse, and that would give us a 20,000 dimensional plot. So at some point, we're going to reach a threshold where we can no longer draw it. And so what we're going to talk about now is how PCA can take 4 or more gene measurements, and thus 4 or more dimensions of data, and make it a 2-dimensional PCA plot. This plot will show us that similar mice cluster together. And we'll also talk about how PCA can tell us which gene or variable is the most valuable for clustering the data. For example, PCA might tell us that gene 3 is responsible for separating samples along the x-axis. Lastly, we'll talk about how PCA can tell us how accurate the 2D graph is. To understand what PCA does and how it works, let's go back to the dataset that only had 2 genes. We'll start by plotting the data. Then we calculate the average measurement for gene 1. So the data is on this 2-dimensional graph. And what we do is we're just going to look at all the x-axis coordinates for the data, and we're going to calculate the average value, and that's that red X. And the average measurement for gene 2. So gene 2 is on the y-axis. And so we're just taking the y-axis coordinates and we're averaging those. With the average values, we can calculate the center of the data. And it's basically, we just draw a line from each average value, and where they intersect is the center of the data. So from this point on, we're going to focus on what happens in the graph, and we will no longer need the original data. So there's our data. Now we'll shift the data so that the center is on top of the origin. So 0.0. So we're going to shift the data. Boop, 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 boop. Bam. So now that X, which represents the center of the data, is sitting smack dab on 0, 0, the origin of the graph. Note, shifting the data did not change how the data points are positioned relative to each other. This point is still the highest one, and this point is still the rightmost point. So the relationship between the data has been preserved. All we've done is just move it. Okay, now that the data are centered on the origin, we can try to fit a line to it. To do this, we start by drawing a random line that goes through the origin. Then we rotate the line until it fits the data as well as it can, given that it has to go through the origin. Ultimately, this line fits best. But I'm getting ahead of myself. First, we need to talk about how PCA decides if a fit is good or not. So how do we even define what best is? So let's go back to the original random line that goes through the origin. And now we're going to talk about how to quantify how good this line fits the data. So what PCA does is it projects the data onto it. So we project it onto, and you'll notice that these projections are perpendicular to the line. So if you're familiar with regression analysis, you may or may not be, but if you are, regression uses a vertical projection where we go vertically up. But with linear regression, we go perpendicular to that line that we're trying to project onto. And then it can either measure the distances from the data to the line and try to find the line that minimizes these distances, or it can try to find the line that maximizes the distances from the projected points to the origin. If these two options don't seem equivalent to you, you're not alone. So what we can do is we can build intuition by looking at how these distances shrink when the line fits better. So we can see as the line fits better, those distances get smaller, and as the line fits worse, they get bigger. So we can see this, like, yeah, sure, those distances get small when the line fits better. While these distances, the distances from the projected data to the origin, those distances will get larger when the line fits better. So watch. You see the projected points start moving further and further out on the line. And as the line fits the data better, and as the line fits it worse, those distances get a little shorter. Now, I'm going to show you how that works mathematically. So to understand what's going on in a mathematical way, let's consider one data point. This point is fixed. So its distance from the origin, oh, excuse me, this point is fixed, and so is its distance from the origin. In other words, the distance from the point to the origin doesn't change when the red line or the red dotted line rotates. So we got this red dotted line and it's rotating, but the distance from the data point to the origin is fixed. When we project the point onto the line, and remember we're doing a perpendicular projection, we get a right angle between the black dotted line and the red dotted line. And that means that if we label the sides like this, A, B, and C, then we can use the Pythagorean theorem to show how B and C are inversely related. Since A, and thus A squared, does not change, if B gets bigger, that's the distance from the point to the line, then C, the distance on the line, must get smaller because we've got this equality and we have to balance that equality. So if B gets bigger, C has to get smaller because A is not changing. Likewise, if C gets bigger, then B must get smaller, again, because A is not changing, and we have to balance the change in C or the increase in C with a decrease in B. Thus, PCA can either minimize the distance to the line, or it can maximize the distance from the projected point to the origin. The reason I'm making such a fuss about this is that intuitively, it makes sense to minimize B, the distance from the point to the line. But it's actually easier to calculate C, the distance from the projected point to the origin. So PCA finds the best fitting line by maximizing the sum of the square distances from the projected points to the origin. And that's a mouthful, and we're gonna step through that one piece at a time. So you'll get to see how we maximize the sum of the square distances from the projected points to the origin. You'll get to see that in action. So here we go. So for this line, so this is that original randomly selected line. PCA projects the data onto it. Then measures the distance from this point, we're going to call that D1, to the origin, or that distance is going to be called D1. And I'm going to keep track of the distances we measure up here. So in the upper left-hand corner of the screen, we've got D1. And then we measure the distance from the next point, so this point to the origin, so that's D2. And then we measure D3, D4, D5, and D6. So we've measured all 6 distances from the projected point to the origin. So here they are. Next thing we do is we square them. And the distances are squared. One reason we're doing that is so that the negative values underneath the x-axis do not cancel out the positive values. It also helps with the math to square things. Because we've got to, this is a side note, not a main idea related to PCA, but oftentimes in data science, you'll see that we're squaring things all the time when we could, in theory, use the absolute value. Now, one of the reasons why we don't use the absolute value is it has a messy derivative at 0. It, in fact, has no derivative at 0. Whereas when we square things, we have a nice continuous thing, and we have derivatives all over the place. So this is an aside that is kind of not to the point, but it's just out of curiosity. You may be wondering why we're always squaring things in data science. And it's just because it makes the math easier because there's derivatives all over the place. Okay, then we sum up these squared distances. And that gives us the sum of squared distances. And we're just going to call that the sum of squared distances. We're going to use a little shorthand from here on out. Now we rotate the line, project the data onto the line, and then sum up the square distances from the projected points to the origin. Boop, boop, boop. And we repeat until we end up with the line with the largest sum of square distances between the projected points and the origin. Boop, boop, boop. Ultimately, we end up with this line. It has the largest sum of the square distances. This line, the one with the largest sum of the square distances, is called principal component 1, or PC1 for short. PC1 has a slope of 0.25. And by the way, a lot of people ask, where does that slope come from? Well, you remember, we started with a random line. And in order to start with that random line, we had to pick a random slope. We already knew it goes to the origin, so the intercept is 0, but the slope was just some random value. And then we started rotating it. And as we rotate that line, in order to rotate it, we have to know what that slope is. So we're not just guessing at what this slope is. We define it. We say, we're going to see what the sum of the squared distances are for a line with slope 0.25. And we go, hey, wow, these are the best. Bam, that's awesome. Okay, in other words, for every 4 units we go out along the gene 1 axis, we go up 1 unit along the gene 2 axis. So the slope of 0.25 is over 4 up 1. And that means that the data are mostly spread out along the gene 1 axis and only spread out a little bit along the gene 2 axis, so relatively little spread on the gene 2 axis. One way to think about PC1 is in the terms of a cocktail recipe or a mocktail recipe. It's really up to you. It's personal preference. Anyways, to make PC1, we mix 4 parts of gene 1 with 1 part gene 2, and then we pour over ice and serve. And it's a refreshing beverage, trust me. Anyways, the ratio of gene 1 to gene 2 tells you that gene 1 is more important when it comes to describing how the data are spread out along PC1. Oh no, it's the terminology alert. Mathematicians call this cocktail recipe a linear combination of genes 1 and 2. I mention this because when someone says PC1 is a linear combination of variables, which sounds very sophisticated, this is what they're talking about. They're just saying PC1 consists of so many parts gene 1 and so many parts gene 2, and this is the recipe for the principal component. It's no big deal. The recipe for PC1 going over 4 and up 1 gets us to this point. We can solve for the length of the red line using the Pythagorean theorem. The old a squared plus equals b squared plus c squared. And we just plug in the numbers, bam, and we do the math, and we get 4.12. So the length of the red line is 4.12. When you do PCA with singular value decomposition, that's what SVD stands for, singular value decomposition, the recipe for PC1 is scaled so that its length is 1. So all we have to do to scale the triangle so that the red line is 1 unit long is divide each side by the original length, which is 4.12. So for those of you keeping score, here's the math worked out that shows that all we need to do is divide all 3 sides by 4.12. This is just sort of an aside. You can come back and look at the video later if you're dying to understand this derivation. I don't want you to think I'm trying to trick you. Anyways, here are all the scaled values. So we've got 1, that line going from the origin to where the X and Y, sort of the over and up part, it's that red line is now 1 unit long. So that's awesome. And the new values change our recipe. So now to make PC1, we mix 0.97 parts of gene 1 with 0.242 parts of gene 2. But the ratio is the same. We still use 4 times as much gene 1 as gene 2. We've just got fancier looking numbers with decimal points. Okay, so now we're back to looking at the data and the best fitting line and the unit vector that we just calculated. Okay, here's another terminology alert. This 1 unit long vector consisting of 0.97 parts gene 1 and 0.242 parts gene 2 is called the singular vector or the eigenvector for PC1. And I'll be honest with you, every now and then, I mean, I made this video a few years ago, but every now and then I have to review this because I'm terrible with terminology, and I can't remember if the singular vector and the eigenvector are the same or not. Anyways, is it scaled? Is it 1 unit? Can it be longer? I don't know. So I can never remember these things, but so this is how I remember, is I actually go back to this and I look at it from time to time myself. What I do remember always is that the proportions of each gene are called loading scores. Also, while I'm at it, PC calls the sum of the squared distances for the best fit line the eigenvalue for PC1. So we've got the sum of the squared distances for PC1 equals eigenvalue for PC1, and that's just some lingo that you got to get used to, because if you ever do PCA in practice, someone's going to be like, what are the eigenvalues? And you're like, oh, well, they're just the sum of the squared distances, no big deal. And if we take the square root of the eigenvalue for PC1, that's called the singular value for PC1. So the thing I always get confused is that the eigenvalue and singular value, they're not the same thing. One is the square root or the square of the other. but the singular vector and the eigenvector are the same. Anyways, bam, that's a lot of terminology you guys just made it through. And I just made it too. So I'm good for like another 6 months of knowing what those terms refer to. It's like getting a vaccine booster, I guess. Anyways, now that we've got PC1 and all figured out, let's work on PC2. Because this is only a two-dimensional graph, PC2 is simply the line that goes through the origin that is perpendicular to PC1 without any further optimization that has to be done. It's just the line perpendicular to PC1. And that means that the recipe for PC2 is -1 parts gene 1 to 4 parts gene 2. And if we scale everything so that we get a unit vector, the recipe is now -0.242 parts gene 1 and 0.97 parts gene 2. This is the singular vector for PC2, and that's also the eigenvector. So the singular vector and the eigenvector are the same. And these are loading scores for PC2. They tell us that in terms of how the values are projected onto PC2, gene 2 is 4 times as important as gene 1. Lastly, the eigenvalue for PC2 is the sum of the squares of the distances between the projected points in the origin, and the singular value is then the square root of that eigenvalue. So hooray, we've worked out PC1 and PC2. To draw the final PCA plot, we simply rotate everything so that PC1 is horizontal. And then we use the projected points to find where the samples go in the PCA plot. For example, these two projected points correspond to sample 6. So sample 6 goes here. Sample 2 goes here. Sample 1 goes here, etc. Double bam. So that's how PCA is done using singular value decomposition, or SVD for short. Okay, one last thing before we dive into a slightly more complicated example. Remember the eigenvalues? We got those by projecting the data onto the principal components and measuring the distances to the origin, and then squaring and adding them together. We can convert them into variation around the origin. Remember the origin is 0, or 0, 0, by dividing by the sample size -1. So I'm just a guest lecturer. I don't know if you guys have talked about how to calculate variation. If not, here's how you do it. The variation for PC1 is just the sum of the square distances divided by n-1, and where n is the number of samples or observations in our data set. And so in this case, we see 6 observations that we measured for gene 1 and gene 2. And so we have n equals 6, and so we'd be dividing by 5. For the sake of the example, imagine that the variation for PC1 is 15, and the variation for PC2 is 3. So we can just look at the graph and we can see that there's more variation along the PC1 because the dots are further apart than they are along the PC2 axis or the y-axis. This means that the total variation around both PCs, so in both dimensions, is 15 plus 3 equals 18. And that means PC1 accounts for 15 divided by 18, which equals 0.83, which equals 83% of the total variation around the principal components. And PC2 accounts for 3 divided by 18, which equals 0.17, which equals 17% of the total variation around the PCs. Terminology alert. We got a lot of terminology we're going through today. So a scree plot is a graphical representation of the percentages of variation that each principal component accounts for. If any of you guys are out there or mountain climbers or rock climbers or something like that, scree So if you have like a mount, like a rocky mountain, sometimes there'll be rock slides, and they just kind of fall down the mountain, and that's that slide, that all those rocks that fell off the mountain, that's called scree. So that's like a mountaineering terminology, scree. And it typically, starts off low and it gets high as it gets wedged up against the mountain. And so these scree plots are supposed to, I guess they reminded someone of what these piles of rocks looked like. So there's another aside. Anyways, we're going to talk more about scree plots later. Bam. Okay, now let's quickly go through a slightly more complicated example. This time we've got PCA with 3 variables. In this case, that means 3 genes. And it's PCA is pretty much the same. PCA with 3 variables is pretty much the same as PCA with 2 variables. You center the data. Oh, by the way, now that we're using 3 genes, now we're kind of faking that third dimension. So gene 3 is depth, and it's going out the back of your computer screen, through the window or the wall or whatever's behind your computer screen. It's going that way. So that's depth. Anyways, just like before, we center the data, and then we find the best fitting line that goes through the origin. We still have to go through that origin, but now we've got a little bit more wiggle room. Instead of just going like this and rotating like a clock, we can now kind of rotate in 3-dimensional space, just like my crazy elbows doing it. So just like before, the best fitting line is PC1. And remember, we define best fitting as it maximizes the square distances between, once we project the data onto that line and the origin. So when we maximize those squared distances, we've got a best fitting line. That's our criteria for defining best. But now the recipe for PC1 has 3 ingredients. So we've got one thing for gene 1, one thing for gene 2, or and then an ingredient for gene 3. So 0.62 parts gene 1, 0.15 parts gene 2, and 0.77 parts gene 3. And so what that tells us is that PC1 the data is mostly spread out along this depth axis, this gene 3 axis. And I'm trying to draw that with these like slightly harder to see smaller balls, and that's like supposed to be far away. And these larger, brighter balls are supposed to be closer to us. And I'm trying to draw that. You just have to imagine it, that we have this 3-dimensional thing going on. So yeah, so in this case, gene 3 is the most important ingredient for PC1. Then you find PC2, the next best fitting line, given that it goes through the origin and is perpendicular to PC1. And a lot of people have trouble with this. You have to imagine that the line right now is going like this, like my arm. I don't know if you can see my arm. And PC2 is going to rotate around like this. A lot of people online, when they look at my video, they're like, that is not perpendicular. But it is, just because we've got some weird 3-dimensional thing. And you can imagine my right arm over here being perpendicular and rotating around my left arm. So that gives us PC2 and the recipe for PC2. In this case, gene 1 is the most important ingredient for PC2. So the data in this case are now spread along gene 1. Lastly, we find PC3, the best fitting line that goes to the origin and is perpendicular to both PC1 and PC2. And I can't do that with my arms. So you just have to take my word for it that in 3 dimensions, we've got one more axis, I guess, going like this. It's kind of a mess, but you can kind of get the idea that in 3 dimensions, it's possible to have 3 different lines that are perpendicular to us, just like the original axes are, but just rotated in a new orientation. Okay, and if we had more genes, we just keep finding more and more principal components by adding perpendicular lines and rotating them. In theory, there is one principal component per gene or variable, but in practice, the number of principal components is either the number of variables or the number of samples, whichever is smaller. And if this is confusing, don't sweat it. We're going to talk about this in about, once we get to the end of this section, we're going to take I don't know, a 5-second break while I stop the recording and then start it up again. And then we'll talk about this in detail. So I know this is going fast now. We'll talk about it more later. Okay. Once you have all the principal components figured out, you can use the eigenvalues. Remember, those are the sum of the squares or of the distances to determine the proportion of variation that each principal component accounts for. In this case, PC1 accounts for 79% of the variation. PC2 accounts for 15%, and PC3 accounts only 6% of the variation. And here's the scree plot. Remember, these are our rocks on the edge of the mountain. PC1 and PC2 account for the vast majority of the variation in the data. And that means that a 2D graph using just PC1 and PC2 would be a good approximation of this 3D graph, since it would account for 94% of the original variation in the data. So we're using the scree plot as a way to justify throwing away PC3 and just reducing it to a two-dimensional graph. So to convert that 3D graph into 2D PCA graph, we strip away everything but the data in PC1 and PC2. And then we project the samples onto PC1. And then we project them onto PC2. And then we rotate so that PC1 is horizontal and PC2 is vertical. And this just makes everything easier to look at. And since these projected points correspond to sample 4, this is where sample 4 goes in our new PCA plot, etc., etc., etc. Double bam. To review, we started with an awkward 3-dimensional graph that was kind of hard to read, and I had to do a lot of arm waving, and yeah, I had to do arm waving, not just hand waving. And then we calculated the principal components. And then, with the eigenvalues for PC1 and PC2, we determined that a 2-dimensional graph would still be very informative because the first two principal components, they account for 94 or 96, I can't remember exactly what it was, over 90% of the original variation. So there's just a little bit of sort of background noise that PC3 accounts for. And lastly, because of the scree plot, we can throw away PC3 and we can use PC1 and PC2 to draw a 2-dimensional graph of the data. If we measured 4 genes per mouse, we would not be able to draw a 4-dimensional graph of the data. Wah, wah. But that doesn't stop us from doing the PCA math, which doesn't care if we can draw it or not, and looking at a scree plot. In this case, PC1 and PC2 account for 90% of the variation. So we can just use those to draw a 2-dimensional PCA graph. So we project the samples onto the first 2 PCs, and these 2 projected points correspond to sample 2. So sample 2 goes here. And then we just say bam, because it's awesome. Note, if the scree plot looked like this, where PC3 and principal component 4 account for a substantial amount of the variation, then just using the first 2 PCs would not create a very accurate representation of the data. Wah, wah. However, even a noisy PCA plot like this can be used to identify clusters of data. These samples are still more similar to each other than they are to the other samples. Little bam. All right, now it's time for a brief pause, and I'm going to stop sharing for a little bit so I can stop recording. Okay, and now, give me just 1 second to get the next thing queued up. Okay, so we're gonna bam. Okay, so note, when we first started to talk about PCA, I mentioned that we were going to talk about it from the perspective of something called singular value decomposition. Oops. Singular value decomposition is the standard way of doing PCA. However, there's an older technique that gives the exact same results based on correlations among the data. So here, I just want to give you a little intuition of how PCA can be thought of in terms of a transformation of these correlations. So with that said, let's say we had some normal cells. These are just, cells from, biological cells. I don't know where they're from, but they're just normal. Psst, if you're not a biologist, these could be people or cars or cities, etc. It doesn't really matter. So even though they look the same, we suspect there are differences. These might be one type of cell, or one type of person, or car, or city, etc. And these might be another type of cell, and these might be a third type of cell. Unfortunately, we can't observe the differences from the outside. So we sequence the messenger RNA in each cell to identify which genes are active. This tells us what the cell is doing. And if these were people, we could measure their height, blood pressure, reading level, etc. Anyways, here's the data. Each column shows how much each gene is transcribed in each cell. Boop, 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 boop. Now, let's imagine, just for now, there are only two cells. We can have, when we just have two cells, we can plot the measurements for each gene. This gene, gene 1, is highly transcribed in cell 1 and lowly transcribed in cell 2. So notice this graph is different than what we had before, where we were plotting the genes were the axes. Now we're plotting all the genes on axes that are for the cells. So that's one difference in perspective when we're talking about correlations and doing PCA versus SVD, or singular value decomposition. That's the primary difference. Okay, and this gene, gene 9, is lowly transcribed in cell 1 and highly transcribed in cell 2. In general, cell 1 and cell 2 have an inverse correlation. That means that they are probably two different types of cells, since they are using different genes differently. Now imagine there are 3 cells. We've already seen how we can plot the first 2 cells and see how closely they are related. Now we can compare cell 1 to cell 3. And we see that cell 1 and cell 3 are positively correlated, suggesting they are doing similar things. That means the same genes that are doing the same gene doing a little amount of work in cell 1 is also just doing a little amount of work in cell 3. Meanwhile, out here in the upper right-hand corner, we've got a gene that's highly expressed in cell 1 and highly expressed in cell 3. So these, in both cells, the genes are kind of doing the same thing. Lastly, we can also compare cell 2 and cell 3. And the negative correlation suggests that cell 2 is doing something different from cell 3. Alternatively, we could try to plot all 3 cells at once in a 3-dimensional graph. So here we've got cell 1 on the y-axis, cell 2 on the x-axis, and cell 3 on the z-axis. Cell 1 could be the vertical, so like I was just talking about, and again, cell 3 is that depth thing, which is kind of hard to imagine, but we're just going to go with it. Okay, we could then rotate the graph around to see how the cells are related to each other. But what do we do when we have 4 or more cells? Draw tons and tons of 2-cell plots and try to make sense of their correlations? Or do we draw some crazy graph that has an axis for each cell and makes our brain explode? No, both of those options are just plain silly. Instead, we draw a principal component analysis graph. So what we've been talking about all along. A PCA converts the correlations or lack thereof among the cells in a two-dimensional graph. Cells that are highly correlated cluster together. So this cluster of cells are highly correlated with each other. And so are these. And so are these. To make the clusters easier to see, we could color code them. And once we've identified the clusters in the PCA plot, we can go back to the original cells and see that they represent three different types of cells doing three different things with their genes. So in this case, we've used PCA as a way to sort of identify sort of unique subsets from the original data. So we just had a bunch of measurements from a bunch of cells, and we didn't really know how to classify those cells. And we use PCA as a way to kind of see if it can make sense, help us make sense of the data. And it did that by compressing the data into a two-dimensional PCA plot. And in this case, it focused more on using the correlations rather than sort of finding that best fitting line. And I'm not a huge fan of using this approach, but people still do teach this way. And when you see PCA out in the wild, they'll say, well, we use the covariances or something like that. And you go, okay, that's fine. That's like SVD, singular value decomposition, but harder to understand. But the results are identical. Okay, so bam. Here's one last main idea about how to interpret PCA plots. This is stuff we've already talked about. The axes are in order of importance, and the more important differences along the second principal axis. Yeah. Okay. Boom, boom, boom. So I'm just sort of repeating what we're doing. So what I'm going to do is I'm going to pause the video because we've pretty much got PCA down solid right now. And we're going to move on to something called multidimensional scaling. And it sounds very scary, multidimensional scaling. But if you understand PCA, or at least the main ideas behind PCA, multidimensional scaling is going to be a snap, and we're going to plow through this really quickly. Okay, so before I get started, I just want to let you know that there's actually two types of multidimensional scaling. There's something called classical or metric multidimensional scaling, and then there's something called non-metric multidimensional scaling. Today we're going to be talking about classical or metric multidimensional scaling. Classical multidimensional scaling is also called principal coordinate analysis, PCOA, which sounds cool, but I think multidimensional scaling sounds cooler. It just sounds so fancy and sophisticated. Anyways, so let's get back to our normal cells. And like I said, if you're not a biologist, these could be people or cars or cities. And like we just saw, even though they look the same, we suspect there are differences. And these could be one type of cell or person or car or city. These could be another type of cell. And these might be a third type of cell. Unfortunately, we can't observe the differences from the outside. So we sequence the messenger RNA in each cell to identify which genes are active. And this tells us what each cell is doing. And we could also measure other things if we were looking at other things. Okay, so here's our data, just like we just had a few minutes ago. Each column shows how much each gene is transcribed in each cell. Boop, 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 boop. And when we did PCA, we converted the correlations or lack thereof among the samples into a two-dimensional plot, or we did it with singular value decomposition, and we projected it into some high dimensional space, and we found that best fitting line, and we called that PC1, and we found the second best fitting line that was perpendicular to PC1. We called that PC2. Okay. So highly correlated samples form clusters. Okay. So multidimensional scaling, MDS, and principal coordinate analysis, PCOA, are very similar to PCA, except that instead of converting correlations into a 2D graph, they convert distances among samples into a 2D graph. And I'll show you what I mean by distances among samples in just a second. So in order to do MDS or PCOA, we have to calculate the distance between cell 1 and cell 2, and the distance between cell 1 and cell 3, and the distance between cell 1 and cell 4. And then we calculate the distance between cell 2 and cell 3, and the distances between cell 2 and cell 4, et cetera, et cetera, et cetera. Okay, for now, let's imagine we only need to calculate the distance between cell 1 and cell 2. One common way to calculate distances between two things is to calculate the Euclidean distance. If we just had two genes, we could draw a line that represented the difference between the values for gene 1. So for in cell 1, we've got 3, and in cell 2, we've got 0.25 as the values for gene 1. And we can do that math, so do that subtraction, and that equals 2.75. And we can draw a line that's 2.75 units long. Then we could draw a line that represents the difference between the values for gene 2. So in cell 1, the value for gene 2 is 2.9, and in cell 2, the value for gene 2 is 0.8, and we do the math, and we get 2.1, and so we can draw a line that is 2.1 units long. Then the Euclidean distance would be the hypotenuse, and that's the Pythagorean theorem. So b squared plus c squared equals the hypotenuse. When there are more genes, we just add the square of more differences between genes. The difference for gene 1, the difference for gene 2, and this is actually a copy and paste error. That's actually supposed to be the difference for gene 3, but it looks a little messy. It should be 2.2 minus 1. Anyways, et cetera, et cetera, et cetera. We just do that for each gene in our list of genes that we have. And ultimately, we calculate a distance. And once we've calculated the distance between every pair of cells, MDS and PCOA would reduce them, all those distances, to a two-dimensional graph. The bad news is that if we use the Euclidean distance, the graph would be identical to PCA. So so we've done PCA 3 different ways. We did it with singular value decomposition. We looked at it with using covariances and correlations. And now we've done it again with something called multidimensional scaling, using the Euclidean distance as our way of measuring distance. In other words, clustering based on minimizing the linear distances is the same as maximizing the linear correlations, or fitting that line so that we maximize those distances between the projected points and the origin. The good news is that there are tons of other ways to measure distance. For example, another way to measure distances between cells is to calculate the average of the absolute value of the log-fold changes among genes. And unless you're a geneticist and working with high throughput data, this may sound completely bizarre, but if you work with genetics data, people are looking at log-fold changes all the time. We'll have we want to see sort of like what's the difference in how this gene behaves in different situations, and we'll say, oh, this gene had a log fold change of 2 or something like that. And that's how we often will kind of talk about changes. So for the example, the log fold change for gene 1 versus, and we're just looking at cell 1 and cell 2. We're not looking at the other cells right now. The log fold change is just the value for gene 1 divided by the value for the value for cell 1, which is 3, divided by the value for cell 2, which is 0.25, and then we just take out the log of that ratio, and that's the log fold change. So the log fold change for gene 2 is the log of the ratio of cell 1 to cell 2. Bam, bam, bam. We just do a lot of those. And then we do that for everything. Okay. Okay, so now we've calculated all of our log fold changes. And then we take the absolute value. So some of those logs are negative, and some are positive, and we're just making them all positive. So they're all greater than 0. Lastly, we take the average of the numbers, and that's the average of the absolute value of the log fold changes among genes. And remember, this is just between cell 1 and cell 2. And so this is the average that we end up with of these values. The average of these values is what we're going to call the distance between these two things. Again, one of the reasons why we're taking the absolute values, we don't want this negative value to cancel out the positive values. We want to have every distance additive and just make it, further and further. Anyways, that's what I just said. Bam. Okay. Ultimately, we get graphs that look different. So on the left-hand side, we've got the MDS plot using a Euclidean distance, and that's the same as our principal component analysis. On the right-hand side, we've got the MDS plot using log fold change. So we had a different distant metrics, and we end up with a different graph. If any of you guys are familiar with there's this thing in genetics called differential gene expression. There's a program called DESeq2 that people use to decide if genes are different from each other. And by default, it gives you, when you put in all of your data, it gives you this graph on the right. It gives you an MDS plot using the log fold change as the distance metric. Okay, as a biologist, we might choose to use log fold change to calculate distance because they are frequently interested in the log fold changes among genes. That's just something biologists are interested in. However, if you're not a biologist or a geneticist, there are lots of other distances to choose from. So for example, there's something called the Manhattan distance. And that's basically, if you just assume you're like in a city with city blocks and you can't go diagonal, you have to kind of go up and over. The Manhattan distance is a sort of up and over distance. And so if you're routing Uber, taxi cabs or something like that, you might want to use the Manhattan distance to get a better sense of how what cars are close to each other, because they could be on the same block, but if there's that diagonal distance between them, that might look short than it really is, because the cab or the Uber guy has to go all the way around the whole block. And so there's different distances for different sort of areas of expertise. And one thing that's nice about MDS is it allows you to use those distances in a PCA-like way. And to be honest, there's no rule for how to select the best distance, and that's kind of the part of the art of data science. Okay, so in summary, PCA creates plots based on correlations among samples or fitting those best lines. MDS and principal coordinate analysis, I'm stumbling all over my words, create plots based on distances among samples. And so PCA correlations, then we do some fancy math, eigen decomposition, or we do singular value decomposition, and out of that becomes coordinates for a graph, and it gives us the percent of variation each axis accounts for. And it also gives us loading scores, so we can determine which variables have the largest effect. MDS and principal coordinate analysis, the only difference is that we can select different distances. So if you understand PCA, just know that the only difference with multidimensional scaling is that we've just, instead of using the Euclidean distance, we're using the Manhattan distance, or we're using this log fold change distance, or whatever kind of arbitrary distance we want to use. But the math, the fancy math, is the same, and the output is also the same. We get coordinates for a graph, we get percent of variation each axis accounts for, and that gives us our scree plot, so we can say, hey, it makes sense to reduce this down to 2 dimensions, or maybe it doesn't. And it also gives us loading scores, so we can determine which variables have the largest effect. So bam. Okay, so now we're going to do another pause. I'm going to stop around. And I'm going to share my screen with you. Hooray. This is, by the way, this is StatQuest Classic. So when I first started doing StatQuest, I used PowerPoint because I didn't know any better. And so this is going to look old school. Okay, so now we're going to talk about something called linear discriminant analysis, also called Fisher's linear discriminant. It sounds fancy. It kind of is, but not really. And we're going to see what it does and then work it out. I'm going to give you a sneak peek. Linear discriminant analysis is good for reducing dimensions. The big difference between linear discriminant analysis and everything we've seen before is before everything we just said, we don't know what we have. Let's just see if we can cluster things in interesting ways. This is different. This time we're going in knowing what the groups are, and we're saying, I want you to separate those as best as possible. So that's what we're going to see here. So here's the problem. We've got a cancer drug. It worked great for some people, but it makes other people feel worse. Wah, wah. How do we decide who to give the drug? Maybe gene expression can help us decide. Yes, I'm a geneticist. I solve all my problems with genetics. So here are the transcript counts for gene X for a bunch of people that took the drug. The green dots represent people for whom the drug worked. And the red dots represent people for whom the drug did not work and it made them feel worse. Wah, wah. For the most part, the drug works in people with relatively few transcripts for gene X. And for the most part, the drug does not work in people with relatively large number of transcripts for gene X. However, there's overlap and no obvious cutoff for who to give the drug. For example, if we put the threshold here, then we would not give these people the drug, even though it would work for them. And if we move the threshold here, then we would give the drug to these people, even though it does not help them and it could hurt them. In summary, gene X does an okay job at telling us who should take the drug and who shouldn't, but it's not great. Can we do better? What if we used more than one gene to decide who should take the drug? So here we have two genes, gene X on the X-axis and gene Y on the Y-axis. And when we use two genes, we can create a better cutoff. Would three genes be even better? Okay, so now we've added a third gene, gene Z on the Z-axis. Remember, that's the one that's giving us depth. The Z-axis is supposed to represent depth, so imagine the axis is going the backside of your computer and into the wall behind it. The big circles are supposed to look closer to you and thus have smaller values for gene Z, and the smaller circles are supposed to look like they're further from you and have larger values for gene Z. Anyway, with 3 dimensions, we can use a plane to try to separate the two categories. To be honest, on a 2D computer screen, it's pretty hard to tell if this plane is doing a good job or not. We'd have to rotate it and look at it from different angles. It's not super straightforward as to how to interpret this plane. And then the question is what if we need 4 or more genes to separate the categories? What do we do then? So when we have 4 or more genes, we can't draw that graph to begin with. So we have the frowny face, wah, wah. And we ran into the same problem when we talked about PCA. PCA, remember that? That was a long time ago. It reduces dimensions by focusing on the genes with the most variation. This is useful for plotting data with a lot of dimensions or a lot of genes onto a simple XY plot. However, in this case, we're not super interested in the genes with the most variation. Instead, we're interested in maximizing the separability between the two groups so we can make the best decision about who's going to get this medication. Linear discriminant analysis is like PCA, but it focuses on maximizing the separability of the known categories. So that's the big difference between LDA and PCA, is that with LDA, we go in with categories that we know are there, people that can take the drug and people who can't take the drug, and we're just looking for the best way to separate them so we can have a nice clean threshold so that when the person goes into the hospital, they can measure their gene expression and go, you're on this side of that threshold, so we're going to give you this drug, or you're on the other side of that threshold, so we're going to give you this, we're not going to give you this drug, and we're going to try something else. And that's how we're going to make a decision about how to treat these people. Okay, so the main idea, again, is linear discriminant analysis is like PCA in that it reduces dimensions, but it focuses on maximizing the separability among known categories. In other words, if we started with this 2D graph and used gene X and gene Y to plot the patients, then LDA would reduce this to a one-dimensional number line that maximized the separability between the two groups. So let's start by trying to reduce the dimensions of this 2D graph so that we have the best way to separate patients. One bad option for reducing the number of dimensions would be to ignore gene Y. And so when we do that, we just project all of the data down onto the x-axis. And this is bad because it ignores the useful information that gene Y provides. Projecting the data on the y-axis and ignoring gene X isn't any better. So we've reduced the dimensions from 2 to 1, but we've done it in a terrible way that is not helpful at all. So LDA provides a better way. LDA uses both genes to create a new axis, and it projects the data onto this new axis in a way that maximizes the separation of the two categories. How does it do that? Now let's talk about how LDA creates a new axis. The new axis is created according to two criteria considered simultaneously. It must try to maximize the distance between the means of the, so we've got two groups, people that can take the drug and people that can't. We're trying to maximize the distance between the means of those two groups. And two, we're trying to minimize the variation, which LDA calls scatter and is represented by S squared within this category. So this is the scatter around the green dots. And this is the scatter around the red dots. I think the reason why they call it scatter and not variation is this thing is really old. This test is very old, before sort of like the language of statistics had been sort of canonized. And so they were just kind of making up terms as they went. That's my theory. That's just a theory. Don't quote me on that. I guess it's going to be in the video. So, okay. So in order to consider both criteria at the same time, LDA creates a ratio of the square differences between the means over the sum of the scatter. The numerator of this ratio is ideally large, and the denominator is ideally small. And so the goal is to find a new axis that can maximize this ratio. Now, to make our lives a little simpler later on, let's replace the difference with D, the letter D for distance. Now, you might be wondering why we don't just maximize the distance between the means. So why don't we just maximize D? Here's an example of why we don't just maximize the distance between means. If the goal is to reduce this two-dimensional graph or data to one-dimensional data or one-dimensional graph by maximizing the distance between the means, and that's all we're focused on, we just want to separate the means as far as possible, then we would create this new axis and project the data onto it, and the means would be as far apart as possible. But the separation of the two groups would not be very good. So let's save this example of just maximizing the distance between the means over here in the upper right-hand corner. Now, if we try to maximize the distance between means while simultaneously trying to minimize the scatter, in other words, we want to maximize that ratio of the distance over the sum of the scatter, then we would project the data onto this new axis. And the means would be a little closer than before. But because we also minimize the scatter, we end up with great separation. Bam. What if we have more than 2 genes or more than 2 dimensions? The process is the same. We create an axis that maximizes the distances between the means of the 2 categories while minimizing the scatter. So here's an example of LDA with 3 genes. So we've got 3 dimensions. Just like before, LDA creates a new axis such that when the data are projected onto the new axis, we maximize the distances between the 2 means, between the 2 categories, while minimizing the scatter. What if we have 3 categories? 2 things change, but just barely. When we have 3 categories, so we've got green, red, and now we've got blue as our new category. The first difference is how you measure the distances among the means. First, you find a point that is central to all the data, and then you measure the distances between a point that is central in each category and the main central point. So those are where our D squares come from. And now we just maximize the distance between each category and the central point while minimizing the scatter in each category. So again, we just want to maximize this ratio, whatever it takes. If we got to shrink some means, and that means we minimize our scatter, then that's what we got to do. Okay, it's the same equation as before, but now there's terms for the blue categories. That's not a big difference. The second difference is that LDA now creates 2 axes to separate the data. This is because now we've got 3 central points for each category, and when we have 3 points, that defines a plane. So if you remember from high school, 2 points define a line, and 3 points define a plane. So remember when we just had 2 categories, we reduced this 2-dimensional or even 3-dimensional graph data to just a 1D line because we just had 2 categories. But with 3 categories, we've got an X and a Y, a new X and Y axes. So with 2 genes, this is no big deal. The data started out on an XY plot, and plotting them on a new XY plot doesn't change anything. So this is not very exciting. But what if we use data from 10,000 genes or 20,000 genes or something like that? That would mean we would need 10,000 dimensions to draw the data. And so suddenly being able to create 2 axes that maximize the separation of 3 categories is pretty cool. So here, this is like real data. Everything I've been showing is kind of like pretend data, but this is a real data that I worked with when I was back at UNC doing research. This is a peptide array. So it's an array that measures how many proteins are in somebody's, I think in this case, it was somebody's blood serum. Anyways, we had 3 different categories, and I applied LDA to these 10,000 genes. And if I just did it if I tried to plot the raw data, I wouldn't be able to do that. Anyways, yeah, so this was in a medical situation. We're trying to figure out how to treat these people. And so that's why you had 3 different categories. These people responded differently to different drugs. And so we're trying to figure out the best way to separate them. And although the separation is not perfect, it's still easy to see that we end up with 3 separate categories. So this does okay. Now, just for comparison, let's look at, we're going to compare this data with LDA and to PCA and see how PCA does it. Now remember, this is LDA, and it does an okay job separating the groups because we're telling it what the groups are in advance. Like we knew that these people, the red people, responded to one drug one way, and we knew the blue people responded to another drug in a different way. Here's what PCA does. Now it takes that same 10,000 dimensional data and reduces it to 2 dimensions, but it doesn't have great separation at all. And it's because PCA isn't even trying to accomplish that. It's just trying to find features that have a lot of variation, and it's trying to find things that are correlated, and it's trying to put those close to each other. It's not trying to find a way to separate things. So the similarities between PCA and LDA are both rank the new axes in order of importance. So PC1 is the first axis that PCA creates, and it accounts for most of the variation in the data. PC2 is the second new axis, and it does the second best job. Just like that, LD1 is the first new axis that LDA creates, and it accounts for the most variation between the categories. And LDA is the second new axis, and it does the second best job, or LD2, excuse me. Both let you dig in and see which genes are driving the new axes. So they both have a concept of loading scores, which are useful for saying, hey, what is causing this separation of these different categories? We can look at the loading scores, and we can answer that question. So in summary, LDA is like PCA. They both try to reduce dimensions. PCA looks at genes with the most variation. LDA instead tries to maximize the separation of known categories. And that's it. So that's it. Thank you guys very much. I know we went over a few minutes. And I really appreciate you guys sticking around and just hanging out with me. I'm good for answering questions. I don't have anywhere to go. So if that's okay, we can do that. It's up to you, and it's up to Shirley, I guess, ultimately.
Thank you, Josh. First, can you stop the recording for now?
Yes.